All right, welcome to The Explainer. Today we're diving headfirst into a world that's, well, it's everywhere. It's on you, it's in you, but you can't see a single bit of it. We're going to be talking about the absolutely fascinating, invisible world of microbes.
So let me ask you something. What if I told you there's an entire universe that exists just beyond the edge of what you can see? I mean, a huge, complex world buzzing with life right under our noses, but totally hidden. Sounds like sci-fi, right? But it's not. This universe is real, and the key to unlocking it, to even seeing it, is called microbiology.
So yeah, microbiology, that's our toolkit. It's our microscope, our map, our guide. It's how we explore this hidden world. Basically, it's the branch of science that's all about studying these tiny living things. You know, the stuff you can't see without some serious help. We're talking about viruses, bacteria, fungi, algae, the whole microscopic shebang.
Okay, so how on earth did we even figure out this world was here in the first place? I mean, you can't see it. Well, it wasn't a single aha moment. It happened over centuries, thanks to some incredible pioneers who slowly, piece by piece, put together this massive puzzle of the unseen.
You know, for the longest time, the idea of invisible life was just, well, an idea. You have Aristotle way back around 350 BCE, laying the philosophical groundwork, just thinking about living versus non-living things. Fast forward a long time, to 1546, and you get Fracastorius, who proposes this radical idea that diseases can actually spread from person to person. He didn't know how, but the idea was out there. Then, things really pick up in the 1860s with the legend Louis Pasteur, the father of microbiology. He's the one who proves definitively that tiny microbes are what cause food to spoil. This is a game changer. Just a few years later, in 1867, Joseph Lister connects the dots and says, hey, if microbes spoil food, maybe they cause infections in surgery. And boom, antiseptic surgery is born. Each step built on the last, leading all the way up to 1928 and Alexander Fleming's famous, completely accidental discovery of penicillin from a bit of mold. And that, of course, changed medicine forever.
So, we've discovered this massive, invisible world. Now what? How do we even begin to organize it? Well, scientists classify everything to make sense of the chaos, and microbes are no different. Let's start with the biggest, most fundamental split.
Right. So the first big dividing line is between two major groups. First up, you've got the acellular microbes. Think viruses. The name kind of gives it away. Acellular literally means not a cell. They don't have that complete, fully developed cell structure we learn about in school. Then on the other side, you have the cellular microbes. These guys do have a proper, well-developed cellulose structure, and this group gets divided even further.
Okay, so within that cellular group, we have another split. You've got prokaryotes on one hand. Bacteria are the classic example here. They have a cell membrane, but their genetic material, their DNA, isn't neatly packed away in a nucleus. It's just kind of floating around inside. Think of it like a messy room. Then you have the eukaryotes, like fungi and protozoa. These are the neat freaks. They're way more complex. They have a cell membrane, sure, but they also have a true nucleus with a membrane around it to hold all the genetic material, plus all these other little specialized parts called organelles. Everything has its own compartment, much more organized.
Now, for today, we're going to narrow our focus. We're going to zoom way in on one of the most common and, frankly, most important groups of microbes out there, bacteria. Yep, those single-celled prokaryotes we were just talking about.
So what's actually going on inside one of these tiny things? Let's take a look. We're going to basically build a bacterium from the outside in and see what makes it work.
All right, starting from the outside, a bacterium has these layers of protection. The outermost layer is the capsule. It's this thick, sticky coating that's great for protection, and it also helps the bacterium cling to surfaces. Right under that, you've got the cell wall. This is a really rigid structure that gives the bacterium its shape and, crucially, stops it from just bursting. And then, the third layer in is the cell membrane. Think of this as the bouncer or the gatekeeper. It's a flexible layer that's super picky about what it lets in and out of the cell.
Okay, let's go inside, past the membrane. The whole cell is filled with this jelly-like stuff called cytoplasm. And floating around in there is the nucleoid. Now remember, bacteria are prokaryotes, so there's no real nucleus. The nucleoid is just the region where all the genetic material hangs out. It's mostly DNA with some RNA and protein mixed in.
Also floating in the cytoplasm are the ribosomes. These things are tiny little protein-making factories, absolutely essential. And you'll also find inclusion bodies, which are basically the bacterium's pantry, little storage pods for food and nutrients.
And bacteria aren't just sitting still. Oh no, they've got parts for getting around and interacting with their world. A lot of them have a flagellum. That's this long whip-like tail made of protein that they use to move, kind of like a propeller. Then on the surface, you see these shorter hair-like things called pili. These are used for attachment, sticking to surfaces, and even sticking to other bacteria to transfer genetic material. And one more cool thing, mesosomes. These are these neat little folds in the cell membrane that create more surface area for chemical reactions like respiration to happen.
So, that's the basic blueprint of a bacterium. But, and this is a big but, they don't all look the same, not even close. Their shape, what scientists call their morphology, is actually one of the main ways we tell them apart.
Okay, let's break this down because it's how scientists identify bacteria at a glance. You've got two main shapes to start with. Cocci, those are the spherical or round ones. And bacilli, those are the rod-shaped ones. Simple enough, right? But it gets cooler. It's not just their shape, it's how they're arranged. So you add a prefix. Mono means it's just a single cell, by itself. Diplo means they hang out in pairs. Strepto means they form long chains like a string of beads. And staphylo means they form these grape-like clusters. So you can put it together. A streptococcus is a chain of spheres. A staphylococcus is a cluster of spheres. This system is absolutely critical when you're looking down a microscope and trying to figure out what you're seeing.
I love this quote from Pasteur. The role of the infinitely small in nature is infinitely great. It hits the nail on the head. So what does that mean in practical terms? Why does all this stuff about tiny cells and their shapes actually matter to you and me?
Well, the scope is just massive. Think about it. In medicine, microbiology is everything. It's how we understand, diagnose, and treat infectious diseases. It's also the basis of immunology, studying how our own bodies fight off these invaders. Then there's industry. We've learned to harness these microbes and use them as tiny factories to produce antibiotics, vaccines, even alcohol. And what about your kitchen? The food and dairy industry relies on microbes to make things we love, like cheese, yogurt, even pickles. And it goes deeper, right into the soil. Agricultural microbiology studies how these tiny organisms impact soil health and help our crops grow. So yeah, from your own immune system to the food on your plate, it is literally everywhere.
So we'll leave you with this final thought. We've just taken a quick peek into this invisible world that's running in parallel to our own. This powerful, dynamic universe of microorganisms is shaping our reality every single second. So now that you've seen a little glimpse of it, how does that change the way you see everything around you?